Bismillahirrahmanirrahim. Assalamu alaikum, my dear students. The topic of our today's lecture is pancreatic enzymes and intestinal enzymes. Objectives of the lecture are to understand the types and functions of pancreatic enzymes and intestinal enzymes. Generally, the different digestive enzymes are secreted in different parts of the alimentary tract and digest different food components such as protein, carbohydrates, and fats. Here it is important to know that these enzymes are released both in response to food and in anticipation like thought of food. Digestion of food starts from the mouth. The first set of enzymes of digestive tract are salivary enzymes, which mainly contains alpha amylases, which break down the starch into maltose and other small polymers of glucose. However, due to the short stay of the food in the mouth, only up to 5% is hydrolyzed. The next set of enzymes in the alimentary tract are the gastric enzymes, which mainly consist of pepsin, which hydrolyze proteins, collagen, helping the other proteolytic enzymes to get in for digestion. Pepsin digests proteins into proteases, peptones, and polypeptides, which are further dealt by the pancreatic enzymes. Stomach is followed by the pancreas, which secrete pancreatic enzymes to further digest the food which enters the duodenum. Chyme in the duodenum stimulates both secretin and cholecystokinin, which are intestinal hormones, out of which secretin stimulate pancreatic bicarb for the protection of duodenal mucosa from the acidic chyme, while cholecystokinin stimulates acinar cells to secrete digestive pancreatic enzymes and also contract the gallbladder. This slide shows the functional histological aspect of pancreatic parenchyma. The pancreatic parenchyma shows different groups of cells. The blue one is pancreatic acinous cells draining into the intercalated ducts, which ultimately opens into major pancreatic duct. This part forms the exocrine part of the pancreas, while the other group of cells in the green color are called the islets of Langerhans, which secrete different types of hormones like insulin, glucagon, somatostatin for glucose metabolism. This part is called endocrine part of the pancreas, which will be discussed in detail in your endocrinology lectures. Today we are concerned with the exocrine part of the pancreas. These blue color acinar cells secrete pancreatic enzymes in the pancreatic juice. They are stained blue because it contains large amount of endoplasmic reticulum, ribosomes, Golgi bodies, large number of mitochondria and zymogen granules. All these take basic dyes. The ductal cells, especially the intercalated cells, secrete large amount of bicarb ions in the pancreatic juice, which is drained in the major pancreatic duct. Major pancreatic duct combines with the common bile duct to open into the duodenum through ampulla of Vater. Pancreatic enzymes include pancreatic amylases, which hydrolyze glycogen like salivary amylases. Lipolytic enzymes include pancreatic lipases, cholesterol esterases, and phospholipase A2. All these enzymes digest neutral fats into fatty acids and glycerol. Proteolytic enzymes include trypsinogen, chymotrypsinogen, procarboxypeptidases, and proelastases. All these enzymes convert protein into tri and dipeptides. Nucleases include DNases and RNases, and some other enzymes include trypsin inhibitors. Apart from the digestive enzymes, pancreas also secretes some inhibitory enzymes like trypsin inhibitors to keep the digestive enzymes in the inactive form and prevent its autodigestion. All these pancreatic enzymes which are released into the duodenum are in the inactive form. In the duodenum, the enterokinases enzymes convert trypsinogen into trypsin, which in turn activates all other pancreatic enzymes. Now, just to revise, carbohydrate digestion starts from the mouth, protein digestion starts from the stomach, while fat digestion starts in the duodenum by pancreatic lipases after bile salts emulsifications. Carbohydrates and protein digestion is also caused by pancreatic amylases and pancreatic proteolytic enzymes, respectively, along with the fat digestion. The next portion of our lecture is intestinal enzymes. Present on the brush border of the small intestine consist of dipeptidases, disaccharidases, intestinal lipases, and enterokinases. Dipeptidases, these enzymes convert dipeptides and tripeptides to amino acids. Disaccharidases includes maltases which cleaves maltose, isomaltases cleaves in isomaltose, saccharase cleaves sucrose, lactases cleaves lactose sugar. All these enzymes convert disaccharides to monosaccharides. Intestinal lipases secreted in very small amount and convert neutral fats into fatty acids and glycerol. The last enzyme is the enterokinases. Enterokinases are secreted by the exocrine gland located deep in the villi. They activate the inactive trypsinogen into active form trypsin. This trypsin then activates all the pancreatic enzymes. Looking at this slide, one can see the activation of pancreatic enzymes trypsinogen into trypsin by enterokinases and the self-activation of trypsin. This brings us to the end of our today's lecture. I hope you have understood the different pancreatic and intestinal enzymes and their functions. Thank you.